My talk is about fungicide resistance in broadacre cropping in Australia, and basically it's about the discovery of fungicide resistance in barley powdery mildew. It's research that's been done both at Murdoch, where I was, and now at Curtin, where we were the Australian Centre for Necrotrophic Fungal Pathogens, and it's funded by GRDC, and it actually involves a whole bunch of organisations, most of whom will not get much of a mention today because of the focus on barley mildew. So I'm going to talk about four things, a little bit about the importance of fungicides, how the risk of resistance depends on various factors, including the fungicide used, the disease used, and the way that fungicides are used, what we can do to manage fungicide resistance, because it is now a reality, and it's as big a part of the scene as herbicide resistance and resistance to genetic methods of controlling diseases, and where we should be going in the future. So the first point to make is that fungicides are become, over the last 10 years perhaps, a really significant part of the pattern of crop protection in Australia into these major diseases. The column here is the potential losses to these various major diseases. You might know this one as Septoria nodorum, tan or yellow spot, stripe rust and barley mildew, just in order of actual losses. So the potential losses is if neither genetics nor fungicides nor in fact cultural controls were in place. So this is the published current actual losses in an average year. And then I just want to make the point that the amount of that is attributed to genetic control, so all that work goes into rust resistance and to genetic resistance to rust and to the other pathogens, is actually rather equal to the amount of control that's attributed to the fungicides. So really, fungicides have become a major part of the pattern of crop protection to major fungal diseases. And some fungal diseases, the ones I've not mentioned here, are entirely controlled by fungicides and not by genetics at all. There is also, so the amount of control given to fungicides is certainly comparable to that attributed to breeding, and in many cases there is growing losses to these diseases, which is, as we can see, we'll see later on, possibly attributed to fungicide resistance. So just to look a little bit more in detail at barley powdery mildew, the losses are almost entirely in the west, so there's a total of $39 million. 33 million is supposed to be lost in the west, and of that, the losses or the savings that are due to fungicides is really significant compared to the potential losses. So barley mildew is predominantly a west issue, and it's predominantly controlled by fungicides. Now, the fungicides that we have registered in this country are listed on cereals, are listed on this table here. And there's a couple of points to make. One is that every one of the fungicides that are registered for cereals contain a triazole. So you may know these as the group C or the group 3 or the DIMIs or even the EBIs or a number of other names. So all of these are all triazole fungicides. The only other group that's currently registered for cereal diseases are mixtures of strobilurins and a triazole. So in fact, every single fungicide available for crop protection of cereals in Australia contains a triazole. And as you'll be aware from herbicide resistance pattern, having a single mode of action is an inherently dangerous situation to be in. Some of these fungicides are actually mixtures of 2 triazoles, others are mixtures of 1 triazole and a strobilurin, as in these 2 cases, Opera and Amistar Extra. So the point to make is that all products contain a DMI, or a ergosterol biosynthesis inhibitor. Some are mixtures, and some are mixtures of DMIs and the strobilurin. Now, when it comes to fungicide resistance risk, there's a lot of experience, particularly from Europe, about how quickly or how easily fungicides lose efficacy because of mutations to resistance in the pathogens. In doing so, we can define a number of features. So in the case of the strobilurins, the risk of resistance, or the frequency that which it occurs, is very high. And that's because there's a single target gene which, if it mutates to resistance, gives very strong resistance. So the degree of resistance, the factor by which the fungus, the concentration of the fungicide on which the fungus can grow, if it's resistant, is in the case of the strobilurians a very high number. It's probably about 100. Whereas in the case of the DMIs, the number here is probably about 5 to 10. In the case of the strobilurians, the target gene is the cytochrome B, and there are single base pair changes in that cytochrome B gene that give resistance to the strobilurian family. And within the strobilurians, there's basically complete cross-resistance. So if you're resistant to azoxystrobin, you're resistant to pyraclostrobin and all the other strobilurians. Across the world, of the diseases I've talked about, strobilurian resistance has been observed in tan spot, or yellow spot as you might know it, Septoria or Staganospora, barley mildew, and the net blotches, the barley net blotches. So basically, everything. DMIs, the resistance risk is considered to be medium, and that's because for perhaps 15 years, fungicide resistance to the DMI group was not observed. What was observed was a slow drift of the effective doses required to control it, because the ratio of fungicide concentration on which the resistant fungi can grow compared with the susceptible fungi is only about 5 to 10. So what you see is not a complete breakdown of control, but a slide towards needing to use higher and higher doses. And in Europe, where there's been no requirement to use particular doses of fungicides at particular rates, what's happening is that growers are now using higher and higher rates that are approaching the maximum permitted allowable doses. The target site is, there's another single target site gene called CYP51, although there are other sites of other genes that mutate to give resistance as well, but this is the major one. And in the world, DMI resistance has been observed, again, in all these pathogens, mildew, net blotch, tan spot, but not actually Staganospora so far, and also scald, the barley disease. So, since 2008, I've been running a project funded by GRDC called the Fungicide Benchmarks Project. And the goal of that project was to isolate examples of these different fungal diseases and determine the level of fungicide that controls those diseases, essentially to get a baseline of control levels in order to be able to be in a position to determine whether any shifts of sensitivity are occurring. So what we've done is, over the 3 years, a group of laboratories in this state and also in South Australia, New South Wales, Victoria and Queensland have collected examples of these pathogens, barley net blotch, barley mildew, wheat tan spot and wheat Staganospora, and sequenced, and we have taken about 100 isolates and put them on 4 or 5 different fungicides and looked for the controlling concentration of the fungicide. And what we've seen is in essentially all cases, there have been some cases of potential resistance, which we have not yet been able to get to a position where we can confirm whether real resistance has been observed in the case of these 3 necrotrophic diseases. In addition to doing the phenotyping, so seeing what concentration of fungicide these fungi can grow on, we've also set out to generate the capability to sequence the fungicide target gene. So we've done that for tan spot, Staganospora and barley mildew, and we've done it for 3 genes, the CYP51 gene, which is the target site for the DMIs, the cytochrome B, which is the target site for the strobilurians, and also the beta-tubulin, which is the target site for the carbendazim types. So the rest of the talk is going to be about powdery mildew, because experience overseas in Northern Europe really has shown that this is the pathogen that first develops resistance. It's got the properties that really suit it to mutating very quickly. In fact, fungicide resistance to strobilurians was observed in the year that strobilurians were released in Northern Europe, in Germany. So barley mildew is a lovely disease to work with. You get these very obvious pustules on the seedling leaves, or also on the heads, and it's over summers as cleistothecia on stubble, so it's all around, and it also infects volunteer barleys that come up before the season gets going. So in order to measure fungicide sensitivity, the slightly more complicated than in other situations because this fungus can't be grown in culture. Instead, what we have to do is get pieces of a barley leaf and insert the two ends in agar. Underneath here, there's agar which has got the fungicide in, either without any fungicide or different concentrations of strobilurin. And the fungicide then translocates through the thickness of the leaf, and we then inoculate these leaves with barley mildew spores, and you are then able to score the amount of disease, either full amount disease or zero disease, across a range of concentrations. And on this basis, we can ask what is the discriminatory concentration, what concentration of fungicide will the fungus manage to grow on. So here you can see at 15 micrograms per mil in the agar, you've got necrotic lesions here which imply that the fungus has tried to infect but actually failed, and the plant has fought back and created a necrotic lesion. There's just a little bit of sporulation here. So we would define this fungal isolate as being controlled by 15 micrograms per mil of strobilurin. What we did in 2009 was we collected about 50 isolates from all around the southern part of the state, and we grew them on strobilurin and triazole, tebuconazole. So each one of these leaves represents a separate isolate. So here you can see an isolate growing happily on 50 micrograms per mil tebuconazole, and an isolate over here growing on 15 micrograms per mil azoxystrobin. And before you ask me what really happened there, we managed to lose that isolate, and so we don't really know what's going on there. But that's life. It's a slightly tricky pathogen to work with. You've got to keep transferring it from leaf to leaf. But with these ones, we did manage to make some progress and discover something interesting. So what we saw was that if we had a range of isolates and we tested them on tebuconazole, and we asked how well did they grow on this discriminatory concentration of tebuconazole, we saw one isolate that really grew very well. And we looked at, so this was suspicious. This isolate we suspected it might be resistant to powdery mildew, and this one we thought would be to tebuconazole, and this one would be sensitive to tebuconazole. So we took that isolate and then tested it more, these two isolates, and tested them more thoroughly. So the sensitive one, we grew it on zero concentration of tebuconazole, and it grows happily. 15 micrograms per mil controls it, and it obviously doesn't grow at any higher concentration. But the isolate that was resistant, it's growing happily at 15, it's growing happily at 20, it's about half inhibited by 25, and it's still growing reasonably well at 30. So this implies that this isolate of the fungus has gained the ability to grow on tebuconazole at perhaps 5 or 10 times the concentration that the wild-type one can grow on. The good news is that that isolate of the fungus, or one which is doesn't have the resistant mutation, it's not affected in its ability to grow on epoxiconazole, or opus, as the fungicide is known as. So both the resistant and the mutant forms of the fungus are controlled at the same concentration of epoxiconazole. And that's actually a major message, that not all triazole fungicides are the same. Although they're the same mode of action group, they're not affected by resistance together. There is not complete cross-resistance. There's in fact a whole range of negative and positive cross-resistances. But what we had here was an isolate which we confirmed to be resistant to tebuconazole. As I said, the target site for triazoles is the ergosterol biosynthesis pathway, and the triazole C class of fungicides target the activity of an enzyme encoded by the gene called CYP51. Which turns iboricol into ergosterol. This is the fungal equivalent of cholesterol, which is why you can eat mushrooms without doing anything with your cholesterol content. The gene for CYP51 has been sequenced a number of times, and there are a range of mutations in this gene which have been associated with resistance to tebuconazole and other EBI fungicides. And what we saw was that the isolate we looked at, it had a mutation at this site C here, whereby an A is turned to a T, which turns a tyrosine at position 136 to a phenylaniline. This is our annotation. So the mutant one has got a single base pair change, which changes a tyrosine codon into a phenylalanine codon. That changes the properties of this enzyme such that it's then able to tolerate higher concentrations of the fungicide. And this is a situation that has been discovered 15 years ago in France and Europe in a number of different powdery mildew species. And this mutation at 136, the Y136F mutation, is consistently associated with strong resistance, so about 10-fold resistance to triazole fungicides in Europe. And what we saw was this isolate had this mutation in, and we didn't actually see any other changes anywhere else in the gene, which suggests that it might have been a single mutation event. What we did in 2010 was we went out again and we collected from all around the southern part of the state. This was, as you'll all be aware, it was a pretty tough year for everything in WA last year, and there wasn't a lot of disease, but we found a disease wherever these arrows are in barley crops. Often they were NVT sites. We targeted NVT sites because they tend to have light fungicide treatments. But we also used a new farm trial site based at South Stirling, so thanks to Scott Payton for being very cooperative in that case. We also sampled from the DAFWA nurseries in Perth here. And what we found was all 50-odd isolates, all of them had this mutation. So we actually were rather disappointed about this because we wanted to look at the evolution of resistance under different fungicide regimes, but from one year to the next we seem to have gone from 1 out of 2 isolates having resistance to all out of 60 or 50 or 60 isolates having the mutation. So this is, I think, deserves some thought and some responses from the community about what's going on here and how we've got to this situation. So basically, we can say that with some confidence 100% of the barley mildew that caused disease on barley in WA in 2010 was resistant to the commoner fungicides. So all the isolates collected from southern WA were resistant mutants. We didn't actually collect any from the northern parts of WA. Mutants are resistant to Tebuconazole, but they're still sensitive to Epoxiconazole. So one question is, does this correlate with field experience? And the second question is, if this is really the case, what consequences are there for managing resistance in the field? So here we piggybacked a trial that New Farm Scott Payton had been running, which looked at propiconazole, tebuconazole, epoxiconazole, and epoxiconazole plus pyraclostrobin, 4 different fungicides, and there was a whole series of complications in this trial, which in fact turned out to be didn't actually aid us particularly. All we could really get out of this trial was that at 3 different concentrations, a quarter dose, a half dose, and a full dose, the propiconazole or tebuconazole was basically hardly touching the pathogen. Really, at full dose, it was perhaps 10% lower than the amount of disease without any fungicide, whereas the epoxiconazole mixture, either with or without pyraclostrobin, Opera or Opus, was fully effective, knocking the disease back significantly, even with just a quarter of the dose, which is what you'd basically expect if it was working as expected. So what we conclude from this is that tebuconazole and propiconazole are basically ineffective even at maximum doses on the condition of that particular trial, which is consistent with lots of anecdotal evidence we've had from the southern part of WA over the last 2 or 3 years. But the epoxiconazole containing fungicides were still effective. And this is consistent with our lab work that said that epoxiconazole is not compromised by these mutations. So that then raises the question, okay, that which of the DMI fungicides, given that all of the fungicides we've got to use at the moment are all DMIs, which of them are actually compromised? Well, here we have a bit of a problem that there's really not much critical data that we can rely on. We have to rely on field performance, anecdotal evidence of field performance, and sub-lab tests. But what we can say from this, I mean, this is really a best guess, is that what we call in the compromised DMIs are propiconazole, tebuconazole, and triademophol, so triademinol and triademophol. So these are essentially the first generation triazole fungicides. Ones we are pretty certain, for a couple of different reasons, are uncompromised and still effective: Epoxiconazole and Prothiaconazole. Prothiaconazole is the major active in Prosaro. But we're doubtful about all the other ones. For example, Ciproconazole, Flutriafol, Difenoconazole, Fluconazole, and, not quite spelled correctly, and Triticonazole. No evidence one way or the other at this moment, but I would put them in the doubtful category. So the question is, how have we got here? We haven't got stocks, samples of powdery mildew barley from the last 10 years, but anecdotal evidence suggests from the South Stirling's area that fungicides, Tebuconazole and so on, have been ineffective or poorly effective in the last few years. I suspect where we've got to is we've had a decade of using susceptible cultivars, Bodan in particular, but cheap but compromised fungicides, and that the pattern of using triazole fungicides 2 or 3 times on the same season, even on the seed, have selected this mutation, the Y136F mutation, and we basically got 100% of the pathogen now is resistant to these commoner fungicides. Now, so if we now look at our compromised fungicides, there's actually 2 different red colours here, it didn't come out. What we can say is that Epoxiconazole and the mixtures of Epoxiconazole and strobilurins are so far okay. There are 2 different reds here, which you can remember that from where we had before. But just before you get too complacent, think, well, I can just use the other fungicides. Well, of course, if Ciproconazole is compromised, azoxystrobin, which is a high risk for developing resistance, is now naked out there in the field. And in case you really think Epoxiconazole is good, well, actually, that's the first triazole that will be removed by changes in EU regulations. So, and also, I would just add that Epoxiconazole resistance has been seen in Europe, but it requires multiple mutations, one of which is this Y136F that we've already seen. So we could have a piggyback situation going on here. So what are we going to do about it? Well, this is the take-home message. Use your integrated disease management practices that you always have done and always should be doing. The take-home message is that DMIs differ in resistance. The first thing to do is destroy your barley volunteers. The second thing to do is to choose a resistant cultivar, and these are the ones from the 2010 list that DAFWA put out that are MR or resistant to powdery mildew, so lots aren't. Avoid the compromised fungicides, which you know are Tebuconazole, Propiconazole, and Triademophon, and if needed, use effective fungicides, so that's Opus, Opera, Amastar Extra, and Prosaro. But please watch out for resistance for these. We can't rely on them being resistant forever. So I think what we've got here is barley powdery mildew is could turn out to be a cautionary tale that should really alert us to the dangers of the practices that we've been using in fungicide use in Australia for the last 10 or 20 years. It's a high-risk pathogen, and we've been using high-risk practices, but it's not the most serious disease on earth, so we can actually live with it, learn from it, and move on so that the other more serious diseases like the rusts and the resistance in those can be avoided. So on a national scale, we need to register more fungicides, particularly from the newer classes that haven't been introduced into this country yet. We need to breed and use resistant cultivars and practice good hygiene. And if we're going to use mixtures of fungicides, we need to monitor the efficacy of both components in a mixture. We also need to legislate the number of successive uses of fungicides, and we need to evaluate the use of mixtures versus alternation. So really what we need to have is a research-based, science-based resistance management strategy, which is just as comprehensively science-based as the herbicide resistance management strategies that have been put in place over the last decade or so. So I'll finish there. Most of this work is done by Madeleine Tucker and Kasia there. So thanks very much.
Just wondering about your testing of it, because we notified the authorities back in 2003, which was the first widespread failure of these fungicides. Okay. We knew they failed because they killed net blotch but not the mildew. So, and spot type net blotch at that. So we had sent samples to the department, later on to you, and just came back nil. and it's not a problem.
Oh, it wasn't in 2003 sent to me.
No, to the department, then later on to you.
Okay.
As in a few years later. But I'm just wondering what's happening between, because we're seeing it in the field, the testing's not showing it.
Oh, okay.
Even now your tests are showing Opus is okay, well, turning down the field Opus just isn't working at all. Jockey's not working, we have to use unregistered or just don't grow barley. But I'm just curious what's happening with the testing, why aren't you picking up what we're seeing?
Okay, number of questions there. One thing is we're still working out methods to actually collect isolates from the field. If you're one of the people who sent us isolates, then we've actually never managed to isolate a fungus off samples that have been sent to us. It's only when we've gone out, collected them and used them straight away that they've been able for us to test. So we're learning as we go here. It's been quite a small operation and we're getting better at it. The second thing is that we collected DNA from the pathogen in 2008 and we recently looked back at then with our PCR test and it was resistant then. So I suspect powdery mildew has been resistant to Tebuconazole for quite a long time. I'm surprised, or let's say surprised you're saying Opus is not working. It's always possible, well, we need to do the assays side by side in a controlled conditions in the kind of lab-based system that I talked about to have some confidence that something's going on. But worldwide, there's no consistent reports that Opus is not working against any fungus. So it would surprise me.
We know it's not working because it's controlling other heart diseases, but not mildew.
Well, whether that's because of a basic, it's just basically not very good for that disease, or another way. Yeah, well, okay. Well, in that case, that's gone as well. But from our results, we would suggest it's still as effective as it ever was. But maybe it wasn't ever very effective. But if it's also not working, that just adds to the worry, gloom and doom.